Our next speaker is from the Sanger Institute. It's Omer Bayraktar. I've probably done terrible things with the name. Please, if you pronounce it wrong, please correct. And he's going to talk to us about exploring human brain telediversity using large-scale approaches, combining spatial transcriptomics, imaging, and functional screening. We can see your slides on the screen. We can't hear you.
Fantastic. Well, thanks, Caleb. Lovely. Thank you. Well, so it's a pleasure to join you guys today. And about my title, I think I was late in actually sending my title. And I think, the workshop organizers nicely lifted it from my website. Our lab really aspires to do all of those different things. But today I'll be telling you mostly about the spatial transcriptomic workflows we've been together to really study cell type diversity in the nervous system. So I think, several speakers gave really, the whole workshop has been this I think excellent introduction and included many great examples of single cell and spatial genomics. For us, the sort of most interesting feature of this emerging field is to be able to combine the strengths of these different methods, different technologies of single cell and spatial genomics to be able to create these multimodal representations of tissue architecture. We are quite interested in doing this in the healthy nervous system to be able to do cell type mapping, both in the human brain, throughout several collaborations that we have at the Sanger Institute and elsewhere, especially within the Human Cell Atlas. We are interested in expanding these to other organs. As part of what I'll tell you about today, we're actually quite interested in providing both experimental as well as computational infrastructure to be able to get this stuff done. So today I'll actually focus most on some of our recent computational work, telling you about how we are developing new tools to allow the computational integration of different kinds of spatial transcriptomic data with single cell sequencing. So I'll start by telling you about our work in the field of spatially resolved sequencing. Again, I think this crew really needs no introduction after several excellent talks, but the very quick primer is that this is really the art of positionally capturing full transcriptomes from thin tissue sections and read them out with sequencing. So this is the kind of technology that really works well in the hands of people who have done single cell sequencing, because first you're getting unbiased transcriptome-wide data. Second, practically, it's quite simple. If your lab runs standard histology or molecular biology, you can get on with it pretty easily. And as you've seen this morning from that excellent talk from BGI, the spatial resolution has just been, I feel like it improves every 2 weeks. So, it started with the progenitor of the technology, spatial transcriptomics. I give you this 100 micron resolution. At the moment, commercially available is the Visium assay, which gives you this sort of 50 micron resolution, which corresponds, depending on your tissue, mostly to 2 to 5 cells per location. And, both in the sort of homebrew and in the production pipeline of commercialization, we have different solutions like slide sequencing that are giving you much higher resolution. So really, I think our ability to do high spatial resolution interrogation of full transcriptomes is increasing. However, even with the high spatial resolution methods, the data that you get from these spatial RNA-seq assays is not exactly kind of like the data that you get from single-cell RNA-seq. And the simple reason for this is that all of these methods are basically grid profiling type of methods. So when you put a piece of tissue section on these arrays, you simply do not get a one-to-one match between a cell and RNA spots. These array spots. With Visium, it's quite, I think, obvious that each of these large spots will contain multiple cell types. But even with methods like slide-seq and even with higher resolution methods, what you end up having is that these small spots will be contributed RNA from fractions of multiple cell types. So right off the bat, the data that you get from these assays is not cell type specific. And to be able to do the kind of things we do with single cell genomics, we actually need computational approaches that can deconvolve cell type specific information from these technologies. So that's precisely what I'll tell you about today. So recently, our lab teamed up with Ollie Stegel, our fantastic computational collaborator from DFK and EMBL, and we developed a tool that we call Selfrelocation that allows probabilistic integration of single cell and spatial omic data. The idea we have here is really to leverage the strengths of both technologies. We start with a single cell data set of a given tissue. This is just processed according to whichever way you prefer. And what we do is that we use the single cell data basically as a cell type dictionary to take our spatial transcriptomic data sets and deconvolve it as cell type specific information. So with that in hand, it allows self-location and really allows you to take all of your cell types in your reference and map them to tissue spatial locations at once. And with that information, you can even do more fun things. You can go about identifying tissue regions in an automated manner based on their composition of different cell types, but then you can also ask more sophisticated questions, such as which cell types are actually co-located. So today I'll just give you a very high level description of the model, but the idea here really is that from the single cell data, we are extracting the average gene expression profiles. We can both sort of use the clustering that you do, but we also provide negative binomial regression models that can actually do this in a manner that negates the effects of axes and everything. And then when we look at spatial transcriptomic data, really the way we've designed self-location, we can work with any kind of count-based data. So Visium, slide seeker, nanostring, as I'll show you examples. And then self-location basically takes your spatial mRNA counts and deduces the sort of the absolute abundance of each cell type from the single cell reference, which gives you their cell type locations. A quick look under the hood, what we're doing here is basically a negative binomial modeling of the spatial counts. An important feature of our model is that we are actually accounting for the technical sensitivity differences between single cell and spatial transcriptomic data. This is actually emerging to be quite an important thing if you want to do this kind of integration work, and it's really required for precise integration of these data types. On top of it, we also have model bits that allow you to sort of take into account additive background signal in spatial data, such as free-floating RNA. And we are also doing slightly more sophisticated analyses, such as taking into account co-location of different cell types, which we find that in bulk spatial data like Visium significantly increases your accuracy. So if you're familiar with these kinds of tools, you probably noticed that over the past 6-7 months, a number of really excellent tools have come out. And I think a lot of them are really like ready out of the box. They give you good data. But with self-location, sort of our specific advantage comes, I think, in our ability to deal with complex tissues a lot better. So with self-location, we've really built it to be able to take very complex single cell references that contain hundreds of cell types and map challenging cell types like rare cells or transcriptionally subtle cell states. I will not show you any benchmarking today. You feel free to look at our Bayraktar paper, which has some, and we'll be adding more. But I can tell you that sort of the number one unique thing with self-location is our ability to take these really challenging, rare and fine cell states and map them more accurately than other methods. And I think this is a really important thing because, I think in a lot of sort of tissue biology questions, generally the most biologically relevant and interesting cell types are these sort of rare and fine cell populations. On top of that, the self-location model, it's also really easily applicable to different sort of spatial technologies. You can easily adapt it, and it's computationally efficient, so you can scale it up to large data sets. So I'll just jump right into showing you some results. So as we developed self-location, we really wanted to have like a model, a really well understood spatial model that we can use to test drive it. And what better model than the mouse brain, as all of you would know. So to create a basically pilot data set for our application, we actually generated a paired single nuclei and Visium spatial transcriptomic data set from the adult mouse brain. So here we have basically applied to consecutive thick and thin sections, either single nuclei or Visium spatial transcriptomics. And we did this at an anatomical level in the mouse where there are many different mouse brain regions. So you expect to get lots of you expect to see like a rich anatomical diversity of different cell types. And I will not show you any of the QC, but the data was quite good, detecting up to almost 10,000 genes with Visium, and also with single nuclei sequencing about, I think, 5,000 to 8,000 transcripts per nuclei. So this is now jumping right into the results. On the left, you can see our single nuclei data, where we have done sort of a broad, not incredibly detailed annotation, where we could annotate almost 60 subtypes covering many of the regional neuronal and glial subtypes. And on the right, you'll see a little animation where we have highlighted 6 of these neuronal and glial cell types that map to known brain regions. And you will see throughout the animation that Selfrelocation will sort of calculate the average gene expression profiles and estimate their abundance across different Visium spots on this mouse brain section. And as you can see, we have successfully mapped these layer neuronal as well as sort of other glial and thalamic neuron populations to where they're supposed to be. So I love showing this animation because I think, it's a bit flashy, but, it shows you really that you can start taking these cell types from single cell data and map them into space. But really the true strength of this method is that we can actually take all of the cell types from our single cell reference and map them in high throughput. So that's basically, I think, really opening the door to doing these sort of cell atlasing applications in a comprehensive tissue-wide manner. So the results of that are shown here. So you can see the anatomical annotation of this tissue section that we profiled on the left. In the middle, what we've done is that based on the sort of estimated abundance of each of these 59 cell types at each Visium spot location, we have actually clustered different Visium spots to automatically actually predict different tissue regions. And those of you who are familiar with mouse anatomy, mouse brain anatomy, you'll see that we have recapitulated nice features like cortical layers, thalamic nuclei to some extent, hippocampal divisions, etc. And with this in hand, what we can really start doing is to really just take a fully automated look. We have our regions here on this dot plot on the x-axis. On the y-axis, you can see all of our different cell types, and you could really sort of quantify the distribution of all of these different cell types at once. So, when we look into this dot plot in detail, we saw many good patterns, both sort of broad as well as fine neural populations, that map to their right locations, homogeneously distributed cell types, at least at the annotation level we did, such as endothelia, that appear in all areas. But sort of one set of results that I'd like to highlight for you is that some new biology that we have found in the heterogeneity of astrocytes. And the way I'll present to you is actually in a way to sell you our method, in that we can identify these rare and transcriptionally subtle cell types and map them to distinct places. So astrocytes, they have a very special place in my heart. I spent a good deal of my postdoc trying to understand their spatial heterogeneity across the nervous system, and I was lucky to work with David Rovich in UCSF in Cambridge in looking at cortical astrocyte heterogeneity. So I was quite excited to just pull the astrocytes from our single nuclei data and see what we got. So when we looked at our astrocytes in our single cell data set, we found that there are actually a number of clusters we could identify using unsupervised methods. We knew very little about the markers that would distinguish them or where they would come from, but one thing that was obvious to us was that there wasn't a great deal of transcriptional heterogeneity between these subtypes. There were relatively few transcriptional differences. But with Selfrelocation, what we could do is that we could basically take our single cell reference, including all these astrocytes, and map them to the tissue section at once. And when we did this, we saw that a lot of these populations nicely mapped to different brain regions, ranging from cortical astrocytes to hippocampal to thalamic and hypothalamic astros. One sort of a question we ask about astrocyte biology, in addition to all this tissue atlasing stuff, is really the level of heterogeneity of astrocytes within each brain region. So, there's actually pretty good evidence from Stan Linnerson and some of David's earlier work that across major divisions of the nervous system, you have these astrocyte heterogeneity differences, but how about within each brain region? And with our data set, we were quite fascinated to see that a couple of the subtypes we identified nicely mapped to subpopulations of astrocytes in the diencephalon. So we could identify this one subtype that we call a hypothalamus astrocytes were nicely enriched in that brain region, while in the thalamus, we're quite surprised to see actually astrocytes mapping to different nuclei. The one population I'll talk about in a bit more detail is this astrocyte, molecular astrocyte subtype we've found to be enriched specifically in the medial habenula, but also in medial versus lateral thalamic nuclei, we also identified molecularly distinct astrocyte populations. And all of our sort of location predictions here, we were able to come in with orthogonal tools like RNA-scope SM-FISH and validate in a quantitative manner. So the plots that you're seeing here, basically, we have performed RNA-scope multiplex FISH against astrocyte markers as well as molecular markers of each of these subtypes. Then we actually quantified RNA expression of these cell type markers at the single cell resolution. And we could easily identify that the spatial distribution of these marker positive astrocytes matched very nicely what you found in the self-location predictions. So the one subtype that I'd like to highlight a bit more here is the habenular astrocytes, because I think it really showcases what self-location can do. So if you look at this astrocyte population at the single nuclei level, it's extremely rare. It makes up almost less than 0.1% of the single nuclei references of the nuclei in the single nuclei reference. In terms of the Visium data, you can see that, in a mouse brain section, which would consist of something like, I think, maybe 2,000 to 3,000 Visium spots, it only, localizes to just a very few Visium spots in the medial habenula. And this is something that we can validate with SM-FISH. So I think what I hope this convinces you that by doing this kind of method, by taking low spatial resolution or relatively low spatial resolution transcriptomic technologies like Visium, if you were to integrate it computationally with single cell data sets by using tools like self-location, you can start doing some pretty detailed tissue mapping experiments that, beyond mapping major cell types, also gives you information on these rare and transcriptionally subtle cell types. So in the second part of the talk, I just want to sort of give you examples of the other kinds of things that self-location can do. So, I'm quite interested in basically applying self-location and spatial genomics in the nervous system, but we are actually quite eager to challenge our model by applying it to different sort of tissues. And I think, some of the different tissues in the human body pose unique challenges. In the brain, we saw many examples of sort of close cellular interactions earlier between, astrocytes, microglia, and neurons, but more or less, in the brain, you have this strong regionalization of the most abundant cell types. So, so if you look at different neuronal and glial cell types, they really localize to different brain regions or sort of cortical layer divisions, etc. But in many other tissues, like immune organs, like the human lymph nodes, you actually have a different story where you can have significant spatial intermixing between different cell states. And when we looked into the human lymph nodes with our self-location, we were quite happy to find that we can actually take single cell data and take a published Visium data set of the lymph node, and we could actually identify both these broad as well as fine immune populations, such as those immune cell states that are different between dark versus light zones or the germinal centers. We could actually nicely distinguish them spatially and map them to sort of find different locations. So it's really, I think, shows you that we can deal with diverse architectures. By being at the Sanger Institute, sort of, standing guard at a, really, I think, leading genomic center, we've also been really lucky to be able to collaborate with other groups and apply self-location to diverse tissue mapping experiments. So akin to the human lymph node, we were able to work with Sarah Teichmann's lab on a lovely paper that they've just had accepted at Nature to actually do single cell and spatial cell type mapping in the developing human gut. And, we could actually nicely see that on Visium data sets generated by the human fetal gut, self-location could take their quite detailed cell type reference containing sort of different cell types ranging from enterocytes and sort of Algier positive stem cells to, more tertiary lymph node type of cells and faithfully map them to different tissue locations. So as part of this work, both in the nervous system and in other tissues, we are also quite interested in directly linking the self-location type of analysis to high throughput analyses of cell interactions. So as part of another collaboration that we had with Rosa Tormo and Sarah Teichmann on mapping the human endometrium, we were actually quite lucky to be able to sort of, take the self-location approach to the next level. So after we performed spatial cell type mapping of these novel epithelial cell populations in the human endometrium by the combination of single nuclei and Visium transcriptomics, we were actually able to use this spatial cell type information to prioritize cell interaction analyses from single nuclei data. And the idea there, I think, as most of you would guess, is that if you perform sort of a receptor ligand expression-based analyses of single nuclei data, you have this really nice throughput of being able to assess combinations of different cell types expressing matching or non-matching ligands, but you have no idea if those cell types are actually in close proximity of each other in the tissue. And with something like self-location, you're actually able to add that dimension. I will not explain this plot, but really the idea here is that after you do your receptor ligand expression analyses of single cell data, you really just focus on the receptor and ligand expressing cell types that are within close proximity of each other in your tissue based on your self-location mapping. And finally, I'll close by telling you about how we are applying self-location now to other technologies than Visium spatial transcriptomics. So the way we built the model really, I think, allows easy extension to other modalities, be it slide-seq or more probe-based methods, as you will see in the next few slides. So an easy experiment was really to take the wonderful slide-seq2 data sets generated by Evan and McCosco's lab, where they have this wonderful data set of the hippocampus and a little bit of the cortex. And we could basically see that if we took our single nuclei reference and map them into the slide-seq data, the patterns of hippocampal and cortical cell types were nicely clear, showing that our method is applicable to different technologies. So slide-seq and Visium, I think, they give you different resolutions, but, they're kind of really, I think, spiritual, like cousins in terms of like how the assay works. You're basically doing transcript capture from the tissues and doing, library generation and sequencing. So one thing we were interested in was actually seeing if we can apply self-location to significantly different types of technologies. And for that, we were happy to actually see that, when you look at methods like nanostring whole transcriptome atlasing, on which we collaborated with NanoString on and put out a preprint recently, we could actually apply self-location to deconvolve very different data types. So with this NanoString WTA assay, the idea is really combining both probe hybridization and NGS readouts. So, the assay relies on basically these 18,000 probes that are hybridized to the tissue. They have these little oligo tags that can be photocleaved from regions of interest, and then they are read out by sequencing. And we were able to basically apply this technique to the developing human cortex tissue that we received from the wonderful HDBR resource in Newcastle here in the UK, where we could sort of segmentally profile across cortical depth different parts of the germinal zones and the developing cortical plate. And with self-location, we could easily take an established sort of a published single cell reference of the human brain and actually map the abundance of different cell types across cortical depth, nicely showing you that different sort of neural stem cell populations, differentiating neurons, as well as upper versus deep layer neurons mapped to the right place. So again, we can really take this method and apply to different technologies. Finally, I'll close by, I think, a fun application that we've been working on in the lab, where we're actually thinking about how, these cell type deconvolution methods, how they can actually contribute to the analysis of image-based spatial transcriptomic data. So I'm lucky to be going right after Malte from Cartana. We've been also fortunate to work with them on applying in situ sequencing to different tissues. And, using some of their new chemistry, we were able to actually generate in situ sequencing data sets targeting about 158 neuronal markers in the human adult motor cortex. And what I'm showing you here is kind of like the standard type of analysis of this sort of data. So when you do all of your sort of probe hybridizations in cyclic microscopy, you go through these steps of basically decoding your probe-based barcodes to end up with basically spatially identified RNA molecules. Then you perform cell segmentation as shown here. You count the number of different RNA targets from your 150 plex panel on each one. Then you perform clustering, and at that point you can also think about doing integration with single cell data. And when you do that kind of analysis, as you can see here, we could indeed nicely identify molecularly distinct astrocytes or fine neural subtypes from in situ sequencing data alone. And it's really wonderful to be able to then look at where spatially these cell types are at really high resolution. Shown here are really just simple illustration of gray versus white matter cell types. And we could also dive deeper into this. Could show you how fine neuronal populations like mid-cortical layer neurons are nicely located to where they're supposed to be in the gray matter. So I think, this type of analysis is really promising, but we've been thinking whether we could simplify it. And one thing we've been sort of working with is really taking the wonderful single cell resolution in situ sequencing data and treating it as if it's Visium. So instead of going through all these steps after we decode our barcodes and read out the sort of spatial position of the RNAs, we simply divide our image up into a regular grid and then run self-location to integrate a single cell reference. And I think that really sort of nicely saves you a couple different steps and will provide, at the very least, a very nice sort of like first line analysis method for all these applications. And nicely, I think, that's working well. So from a single nuclei reference that was rather broadly annotated, you can see here that we have, sort of divvied up the in situ sequencing data sets into a 100 micron grid. And we have we have estimated the distribution of all these layer neuronal as well as glial cell types. So with that, I hope I illustrated to you that sort of the integration of single cell and spatial RNA sequencing is really a high throughput method that allows you to do complex tissue mapping. And I think with the applications I showed today, I hope enforces the view that you could sort of work with rather relatively low spatial resolution data and still come up with a high resolution atlas by doing this integration. So if you're interested, our tool and our preprint, they're all online. Feel free to check them out. And to close, I think just to give you a sight of sort of like the two future directions we are working on at the moment. The first thing we're thinking, looking at all these different Visium data sets from other different tissues, we're just starting to realize that all of the problems we dealt with single cell biology, single cell transcriptomics, they're still there. They're also here for spatial transcriptomics. So, I think for doing, Visium analysis or other sort of sequencing-based analysis of different tissues, we're going to start to need relatively good batch integration methods that can put together many sections or samples. And finally, as you saw earlier, there are all these wonderful technologies that are just pushing the resolution, allowing you to look at larger samples. So, and that computationally, that's going to be a challenge because it's going to really start to create millions of RNA spots. So one area that we're very much actively looking into, and we're looking forward to share some new developments soon, is sort of a large-scale version of self-location that really helps you to take it to the next level. So with that, I'm a bit over time, so I'll just acknowledge the lab and our wonderful collaborators, including our industrial collaborators. And thank you for your attention. Love to take your questions.
Great, thank you very much, Omer. So I just encourage anyone who has questions to please put them in the Q&A box, or panelists put them in the chat. So we have a first question that's come through. This is from Caleb. And Caleb asks, is the astrocyte gene expression pattern correlated with the dominant local neuronal type?
That's a great question. In the case of the astrocytes that we identified, the sort of like these fine regional astrocytes in the diencephalon, the answer was no. There's a wonderful paper from, I think, the Lopez Bendito lab, where they actually did identify what Caleb was talking about, where they say that if you were to look at more broader regions, such as cortical versus thalamic astrocytes, you can actually identify basically shared developmental gene expression signatures. And that makes a lot of sense, because in most brain regions, neurons and astrocytes are derived from the sort of same pool of neural progenitors. So you would expect there's a developmental patterning. I think what that needs is probably I think it needs a revisit in terms of looking at fine astrocyte subtypes. And one sort of toy hypothesis is that maybe at the sort of big regional level, you have those shared developmental signatures, but when it comes to these fine patterns, such as habenula or thalamic nuclei, maybe they're driven by other mechanisms, be it intrinsic or maybe actually driven by extrinsic cues from neurons.
Great, thank you. And I mean, I was thinking along the same lines as well. So these astrocyte subtypes, how do we have any idea of how the function changes between these subgroups of astrocytes, in particular when you think about the tripartite synapses and the role that the astrocytes have at the synapse? Any idea about that, or how you would look into that in more detail?
Absolutely. Yeah, so I think, we were pretty bland with our sort of, I think speculations of the functions of subtypes in our paper. So I think in the case of these diencephalic astrocytes, there weren't any sort of screaming synaptic regulators or whatnot, though there are some sort of interesting candidates in terms of perhaps interactions with vasculature. One thing we suffered with in this particular analysis, to be completely open with astrocytes, is that, I think, our sort of transcriptomic depth of astrocytes was a bit lower than neurons, and that's kind of, I think, an issue of just doing unbiased analysis. But B, I think for doing more kind of unbiased cell interaction type of analyses, actually, we don't have very sort of richly annotated lists of mouse receptors and ligands. So if you were to look at some of the immune organs that I showed you, there are really these wonderful tools like CellPhoneDB that have these long manually created lists of immune protein interactions. But in the mouse brain, both the combination of it being the mouse over like human, but also the fact that I think a lot of cell interaction type of stuff comes from immunology, not from neuroscience, you don't have lots of information. In the cortex, where we put out a story on cortical layer diversification last year, which we kind of left it exactly at this place. We just said these are the interesting genes. And what others like Nikki Allen were able to show later on was that actually some of our, say, layer astrocyte enriched markers were indeed involved in synaptic maturation of very specific circuits. So I think it'll be a very rich area to look at.
It's very interesting. Lovely. Thank you very much. In the interest of time, we're going to move on.